Welcome to this introduction to the PGC Major Depressive Disorder Working Group. We're going to give you a brief introduction to the group chairs, to what it is we do, to some of the key people in our working group, and some of the discoveries that we're making at the moment. First, let's meet our chairs.
Hello, my name's Cathryn Lewis, and I'm a scientist and a statistician from King's College London.
My name is McIntosh. I'm a psychiatrist from Edinburgh, and together with Cathryn and Lewis, we chair the PGC Major Depressive Disorder Working Group. Our working group works to identify the genetic basis of depression by looking at molecular changes in the DNA of people with and without the disorder. And we do this through the use of genome-wide association studies in the main. We collect data from a number of groups, from a number of people who've conducted genome-wide association studies of their own, and we combine those studies into one large meta-analysis. Let's start first by discussing what it means to have depression. Well, people use depression in two main ways. The first way is to describe depressive symptoms, and the second is to describe an episode of major depression that is clinically significant and might lead to treatment by a medical practitioner. So depressive symptoms are very common. They include low mood, loss of enjoyment, poor appetite, or sometimes increased appetite, poor sleep, sometimes increased sleep, tiredness and loss of concentration. Sometimes people may be troubled by feelings of hopelessness. They may feel very negative about the future, and they may wish for their own death or seek to hasten it by conducting acts of self-harm or attempts on their own life. Major depression or clinical depression is when you have 5 or more symptoms for 2 or more weeks. They affect your ability to lead your usual life, and they cause you suffering. All of our group members are motivated by a desire to improve the treatment and outcome for people who suffer from depressive disorder. We will use genetics because we want to learn about the causes of depression, and genetics gives us a particularly strong route into those causes. It also provides us with the tools to identify the cells, processes, and mechanisms underlying the condition that might lead us to new insights and new ways of improving outcome. We would like also to learn to predict depression from genetic data so that we could identify the people who are at highest risk long before they become unwell so that we could make timelier interventions. And finally, we would like to find better treatments and improve outcome. And genetics provides us with the toolkit to identify treatment targets and to repurpose or develop new treatments that will be more effective than the ones that we already have. We work with more than 200 scientists, psychologists, and psychiatrists from around the world. They collect blood samples and saliva from people with depression, and then they extract the DNA from those individuals and genotype them. They then share their genotype data with us in the PGC MDD working group. We act to collect that data from our members and combine it in a large analysis that includes data from many samples across the world. We then complete an analysis and publish that in a journal, acknowledging the full group membership and all the individuals who've played a role in that work. I'd like to introduce 3 more key working group members: Lu Yi, Fabian Straight, and Carmel Choi. Lu Yi makes the data we have available to other members of the group so that they can conduct their own analyses collaboratively and with the full approval of all group members. Fabian identifies new studies for the next main meta-analysis of PheWAS studies, and Carmel Choi ensures we have good dissemination and outreach from the group, and she represents us on the PGC Outreach Committee. So having discussed the background to the working group, I think now is a good time to turn and focus on what we've learnt about depression through our efforts. So I think when people first started conducting genome-wide association studies of depression, there was an anticipation that we would start to find genetic factors straight away. This proved not to be the case, and we didn't really make significant advances until we started to include our studies together in large meta-analyses. The first one of these is published in 2013 in Molecular Psychiatry and included 9,000 people with depression and identified no genome-wide significant findings. Now, while we didn't discover any genetic loci for depression in this study, it enables us to calculate how many people we would need in order to make those first discoveries. And we discovered that it was going to be approximately 40,000 individuals would need to be included in order to make our first advances. And so this has proved to be the case. In the next PGC publication from 2018, that's published in Nature Genetics, we included clinically ascertained samples, but we were also able to include data from the commercial testing company 23andMe, who do direct-to-consumer genetic testing. Using this additional source of information, as well as our clinically collected cohorts from psychiatrists and psychologists, we were able to increase the sample size to 135,000 depressed people and identify 44 genome-wide significant findings. And then less than a year later, we added to this data from UK Biobank, a study of 500,000 people in the UK who were ascertained for the presence of depressive symptoms. And by combining them and publishing this work in a paper in Nature Neuroscience in 2019, we were able, in nearly 250,000 depressed cases, to identify 102 genome-wide significant findings. The availability of genetic data from our consortium enables us also to test whether depression shares genetic risk factors with other traits and disorders. Now, we found that indeed depression does share genetic risk factors with other traits and disorders, and none more so than measures that look at the genetics of depressive symptoms. In other words, the individual symptoms that make up depressive disorder, but which are present commonly in the general population and measured using questionnaires and other scales. It's also very highly correlated with the genetics of neuroticism, which describes a tendency for people to experience negative emotions, but is a normal personality dimension. It also shares its genetic architecture with a number of other psychiatric disorders. And shown on this graph is major depressive disorder or other studies of major depressive disorder taken from independent PheWAS studies, and also symptoms such as insomnia, attention deficit disorder, bipolar disorder, and schizophrenia. Depression also shares its genetic architecture with what we call modifiable or potentially modifiable environmental risk factors, and these include smoking and obesity, and some of the consequences of these disorders, including lung cancer and body fat. And last but not least, we find that the genetics of depression overlaps with the genetic determinants of the onset and end of menstruation, with education, years of schooling in particular, and college completion, and with expected lifespan. The availability of genetic data also enables us to connect the genetic discoveries and depression to various aspects of its biology that might provide us with the clues as to how depression is manifest in the brain. We find that the genes that are identified from our genome-wide association study tend to be those that are enriched in the brain and that are expressed in the prefrontal cortex, those that are expressed in neurons, and those that are expressed at the electrical and chemical connections between nerve cells known as synapses. Another way in which we can use our samples is to begin to profile people's risk of depression in other as yet unseen samples. So we start here with the Genome-wide Association study from the Howard 2019 analysis, and we can use this information, knowledge of each marker across the genome and how much risk it is associated with, to take a new sample of individuals, here represented by different genomes with different letters, and we can use the weights calculated in the GWAS analyses with the data, the new data that we have, to multiply the risk factor by its effect size to get a polygenic risk score. And the polygenic risk score is a measure of how much genetic risk you're at of depression in an unseen individual. And you can think of it as a bit like a quantitative measure of how much risk you're at that's fixed from birth, but which varies greatly in the population from people who are in the top 10% to people who are in the bottom 10% of risk for a population as a whole. That information collected in a different sample can then be used to see how well we're doing in identifying depression's genetic risk factors. And in this example, we took the results of a previous GWAS analysis and we took it to a new sample called Generation Scotland, and we measured the polygenic risk of everybody in that study. And we ranked everybody from highest risk to lowest risk to see how much risk of depression they were at. and how our measured genetic risk from the genome compared to whether they actually manifested depression at some point in their life. We discovered that we were able to explain about 2.6% of the variation in liability to depression in a population. Or put it in a different way, we were able to show that the people who were in the top 10% of the population in terms of risk, when compared to the lowest decile of risk, the lowest 10%, were just under 3 times more likely to develop depression, which I think demonstrates that we are making progress in better understanding the genetics of depression in independent samples, and that eventually we might move to more accurate prediction that in the more distant future will provide clinical useful information for people who have not yet become unwell. Another way in which our group has used polygenic risk scores is to identify other traits that are associated with depression, in this case traits that were measured in the large UK biobank study of nearly 500,000 individuals. Now, much of this slide repeats what I've shown before, that the genetics of depression overlaps with neuroticism, with depressive symptoms, and with other psychiatric conditions. Some novel findings that were shown in this study were that depression is associated genetically with many brain imaging factors. Now, many brain imaging studies have been conducted with a view to understanding the mechanisms of depression, but by having genetic data available, this helps us to understand if those factors are likely to be associated with the etiology of depression or are likely to be a consequence. And in this paper published by Shen et al. Earlier in 2020, they were able to show that genetic risk factors for depression were associated with many brain imaging variables, and then using another technique called Mendelian randomization, were able to show that the majority of these brain changes are associated with the consequences of depression and its treatment, rather than being associated with its cause. There are many outputs from our group, and on this slide here I've shown the two main most recent meta-analyses, the one by Ray in Nature Genetics in 2018 and the one by Howard in 2019. Now, these provide excellent source reading if you want to learn more about our group and the many papers that have come since. The paper on the far right-hand side of this screen was published in Neuron in 2019 and gives an overview of current progress in uncovering the genetic architecture of major depression. I've spoken so far mainly about PheWAS studies of depression, but we have many other projects on the go. We have a study that looks at the genetics of treatment response so that we can understand the mechanisms of treatment response, develop better antidepressants, and predict those currently most likely to respond to currently available treatments. A number of studies that examine the genetics of depression in African and Asian ancestries that have been traditionally neglected by genetic studies. Also conducting genetic studies of postpartum depression. If you're interested in any of these studies, in providing data or in joining the working group to contribute to analytic methods or to other initiatives, please get in contact with us. If you want to get involved, please reach out to one of our group chairs, either myself or Cathryn Lewis, and you can do so by visiting the webpage shown on the screen now, where you'll find details of how to contact us. So finally, I just want to thank everybody for listening to this talk today, and I hope it's motivated you to become more involved with the activities of our working group. Thank you. Goodbye.